we start looking at the DNA structure, you have your phosphate here, you have your 5-carbon sugar here, and your nitrogenous base there. And on the handout, if you look at the nitrogenous bases, they're split into two. There's purines and pyrimidines. The purines have double rings, and the pyrimidines have single rings. And no, you do not have to ever memorize the structure of these. I would never ask you to do that. When we look at this DNA structure, though, when you count out your DNA, the sugars, that's carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Okay? And when you build DNA, you add from carbon 3 to carbon 5 or carbon 5 to carbon 3. So when you build a new DNA molecule, the phosphate would go here with the carbon, and let's put a G there. Okay? So when it joins, how do we join molecules in biology? Guys? Yes, dehydration synthesis. So when we do dehydration synthesis, I'm going to do the magic of dehydration synthesis. I'm going to take this H and the O, and then an O that comes off here, blah, 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 make our water, and lo and behold, magic happens, and you end up like that. Do you see this? Yes? So we went from one, now we've added another. Is that good? The other side, if we wanted to build on it, what has to be here? A C. And then here you would get your sequence added. And remember, when the A and the T meet, we call that a base pair. Okay? So a base pair has two nucleotides. But what have you noticed about the two nucleotides? What do you notice? How did I write them?
One is inverted from the other.
One is inverted from the other. So one runs this way, and the other one runs this way. They run opposite of each other. In fact, they run parallel to each other, but opposite. So what is termed as is it runs anti-parallel. And when you look at your DNA structures, stop writing just for a second, take a look at this. Just stop writing, look at the screen. When you look, the 5-carbon is free on that top, right? That carbon is free. On the bottom here, it's the 3 that is free. So the 3' end is this end, and the 5' end is that end. Prime meaning the carbon in the 5-carbon sugar. So if you were to add to the top here, you would add to the 5' end. If you add to the bottom, you would be adding to the 3' end. The other side, what's free? The top has what free? Which carbon is that? It's the 3. And the bottom at the bottom is the 5'. Ask me 2 questions. Go. And what holds a whole DNA ladder together is the hydrogen bonds between the A and the T, and the hydrogen bonds between the G and the C. And A and T have 2 hydrogen bonds holding them together. C and G have 3 hydrogen bonds holding them together. So why does DNA have to replicate? To make more cells. Because why the DNA is the instructions. It's the code. So it's kind of funny, they talk about coding. Well, coding happens to be like coding of a computer. You decipher the code to build something, yes? Well, coding for DNA is just deciphering the code and building proteins. Okay? So it's very similar. So if you want mitosis to happen, you need replication beforehand. So if we have a strand of DNA, we need to take this strand of DNA and make a copy of it so that you will end up getting a second copy that is identical. Okay? So DNA replication is making this copy of itself. Okay? And Watson and Crick, you know those fun people, Watson and Crick? Who were Watson and Crick? James Watson and Francis Crick. They were the ones that discovered the double helix of nature of DNA. Yes, they discovered, I'm going to put discovered in quotations. They discovered this, and from that they figured out, oh, from there, replication of the DNA makes complete sense and why it doesn't make mistakes. It's challenging when you think about having to make 3.1 billion of these lines and copy it exactly the same. And think about it, in 9 months' time, or 40 weeks, your cells have gone from one cell with 3.1 billion base pair to billions or trillions of cells with those same 3.1 billion base pairs exactly copied. That is just remarkable to think that that can happen and that it doesn't have as many errors as you would think it would have. Okay? So Watson and Crick came up with this theoretical double helix and made it practical. How did they get it? Who helped them? Who should have got the Nobel Prize? Rosalind Franklin. It's Rosalind Franklin that got it for them. Look at how DNA replication works. This is just a quick look. I'm going to look at it from a very basic level. Tomorrow or the next day, we can do it very detailed, or you can do it on your own if you'd like. But here's the base behind it. Number one, you need an enzyme that unzips the helix and unzips the bonds. So see how it splits open? So I'm going to make that the triangle. So there's an enzyme that unzips the helix and unzips the bonds. Okay? Number two. Next thing is you need to prime the DNA. Why would you prime paint? Why do you have to put primer on the wall? So it sticks better, exactly. So, why do you have to prime the DNA? To ensure the enzyme that reads it sticks to the DNA. Okay? So the second step is to prime it. So I'm going to put the primer in red, and it's basically just a sequence of DNA. Okay? Number three, I'm going to add an enzyme that reads and builds. And so it's going to read the template and it's going to build the complement. What's it building? What is this strand called? Sorry? Simple strand. What's it called? What are we dealing with? Okay, well, this is a template of the parent strand. But what's this big thing called? The regular. We're talking about it. Starts with D, runs with N, has a name. DNA, yes. It's DNA. And we're building a polymer of DNA. So the enzyme that builds a polymer of DNA is DNA polymerase. So DNA polymerase is the enzyme that builds it. Who knew? So the DNA polymerase has to jump on at the primer. There's my DNA polymerase. It's going to jump on and it's going to start going this way and building a new strand. Easy? Here's a little caveat. It's like you guys. It only reads 3' to 5', which means it builds 5' to 3'. So see when it attached here? It started at the 3' reading and keeps reading to the 5'. So this new DNA strand will build, and now you're going to get the DNA building. What's wrong with the top strand? So up top, you have to not start here. You can't start there. What you have to do is you have to throw the primer in further downstream, and then have polymerase attach here, and it'll go backwards. Okay? And then further downstream, another primer will attach further down, and polymerase will build again. Okay? And so what you get is what's called a leading versus a lagging strand. And I'm done.